greenhouse crops, because until now, it was all about growing in the field. There was some horticulture in the previous session, these peach trees, but no greenhouse tomatoes yet. And that's what I will talk about. Actually, it's work from our PhD student, Yutaka Tsutsumi. She is back in Japan as a postdoctoral researcher, and she worked together with Fred, George, and me, and also Daniela and Martin were involved. So I'm happy to see that they are all present here. But there were a few more people involved who are not present today. Well, what I have here in this sheet, it was mentioned already by several presenters before. Yield is an important target for breeding, but it's also difficult because it's a complex quantitative thread with many genes and a strong interaction between genotype and environment and even management. And traditionally, with selecting on breeding, we can get quite far, but in what I propose here in this 15 minutes is that in an alternative way, perhaps we can get even farther, where we select not on yield itself, but on underlying components. And then we have to bring from these components, go back to the level of yield. We can use crop models. And I say crop models here, but in the example I will present, that is still a bit limited because that's yield-dissection models. So that's static models, but I will show at the very end that we can also do this with dynamic models. So the 5 steps in the procedure are shown here. So we dissect yield into yield components. Then we identify the genetic basis for these components, the QTLs, and then we build a prediction model for these yield components. And then 2 more steps. These predicted values for the yield components, they are inserted into the crop model to predict yield. And that model, as I said already, that can also be a dynamic model, including environmental factors, but that's not in the example I show. That's a static model. And then the last step, we use the crop model for the ranking and selection of high-yielding genotypes. What we expect is that we get, in this way, more QTLs available. We get an understanding of QTLs for yield. So, for example, a yield QTL, we can see probably if that is a yield QTL because it influences the harvest index or because it influences the photosynthesis rate. We get more stable QTL, at least that's the expectation. And some of these underlying components, maybe we can determine already in an early crop stage. To give you an idea, for high-tech tomato cultivation in the Netherlands, the cultivation takes 11 months. So if we can do our analysis over a shorter period, that would be very helpful and economically speaking, meaningful. So the aim of the work with 2 tomato case studies is to determine the direction, so both as component threads and as QTLs for improving the yield, and to identify the phenotypic and genotypic idiotypes for a high-yielding cultivar. And as said, there were 2 case studies, one in Spain, where we had 346 rills in a population with 3 parents, 1 elite parent and 2 wild relatives of tomato. And in Spain, the cultivation was done in 2 greenhouses at 2 locations, both around Almeria. And you will see in further slides that I refer to Fernando and Juan Manuel. You guess already that's the first names of the farmers of those greenhouses. And the cultivation was from September till May, so typically a season for Almeria. And then there was another population which was grown in the Netherlands, 342 rills of a crossing population with 4 parents, 2 elite lines and 2 wild species, and grown in the Westland area from February till November. And that Dutch study has already been published a few years ago in silico plants. But the Spanish study is in the thesis of Yutaka. I will refer to that at the end, but not yet in a journal paper. Well, Fred showed yesterday also this slide. We used 2 yield component or yield dissection models. One where we focus on the sink, and they're, well, very basic, just like what many grain people do or wheat growing people do. We dissect the yield into the number of the organs, so the number of fruits, and the individual fruit fresh weight. And the other dissection model is focused more on the source. So we call that the biomass dissection model, where yield is seen as the product of total dry matter production in the fruits, so the total dry matter which is allocated to the fruits over the whole season, multiplied by the fruit fresh dry weight ratio. That will give the fresh fruit weight cumulative, so that is the yield. And then this total fruit dry matter, again, can be seen as the product of total biomass multiplied by the harvest index, the fraction which goes into the fruits. And then the last step, this total biomass, we can see that as the product of cumulative light interception over the season multiplied by a light use efficiency. Well, first some introductory slides about our data and our assumptions. So you expect, at least we expected, that if we look at yield components, that there is a higher heritability than for yield itself. That's the whole idea behind this section in components. And indeed, that was the case. Here I show the results for the two Spanish greenhouses. For yield, heritability was quite low, 0.12 or 0.30 in the two greenhouses. And if you look at components, well, it could go up to 0.65 as for the individual fruit fresh weight. So clearly, this first point, what you expect, higher heritability for components, so closer to the genetic basis, indeed that was the case in our data set. And that also resulted in more QTLs for the underlying components. For yield, there were 7 or 9 QTLs found in the data sets of the two Spanish greenhouses. And for the underlying components, yeah, it was 9 or higher, and the highest was for the number of fruits per truss, where there were 17, just the same as for the individual fruit fresh weight, 17 QTLs were detected. So also that fitted with what we expected and certainly had hoped for. Well, what was also, well, I would say for me quite surprising, because in the Dutch study, the rills, well, there were some rills with a higher yield than the elite parents, but not too many. But in the Spanish population and in the Spanish environment, remember I talk about Dutch and Spanish, but that's not only the location, that's also the set of rills, which was different. So it's always the combination of these two. But in these Spanish experiments, there was about 50% of the rills had higher yields than the elite parent. And I think that's exceptionally high. Also, if you consider that the integration came from wild tomatoes, and if I look to wild tomatoes, they look very poor. They don't grow very well, they have very small fruits, but apparently there is genetic information which can boost the yield of elite lines. And how much higher it was, that's also a quite big step for both greenhouses, more than 25% higher yield for the best performing rill compared to the elite parent. But then the question is, which component traits caused this yield improvement? And then if I start with the harvest dissection, well, we found that both the individual fruit fresh weight and the number of fruits produced per plant were positively correlated with yield. However, those two components were negatively correlated. And that makes it difficult to directly breed for higher yielding. If you focus on, for example, the number of fruits to improve that, most likely that will lead to a lower individual fruit fresh weight. And that's also shown here in this graph. Let me see, well, yeah, this works. So on the x-axis, we see the number of fruits. On the y-axis, the fruit size, individual fruit size. The cross of the orange line, that is where the elite parent is situated. And every dot is one of the rills. And then we can conclude that, well, here a lot of dots is here in this quadrant. So there we have higher number of fruits, but that goes together with a lower individual fruit weight. And we can also see that rills which result in a higher yield. So I should explain these blue lines, that are the combinations of equal yield. So the elite parent is here on this solid blue line. If we go a little bit higher with dashed blue lines, that most of the rills which have a higher yield, that is caused by rather by more fruit than bigger fruit. Of course, there are also some which show the other way around, but the majority is higher yields than the elite parent because of more fruits produced. Well, then I have similar graphs, but a bit smaller, so I hope you can still still read, for the biomass dissection, and then for the three conditions we had, so the two Spanish greenhouses and the Dutch greenhouse, so two Spanish and the Dutch, and then on the top we plot the total fresh dry weight, the total fruit dry weight, sorry, against the fruit fresh dry weight ratio. So that's the two components here in the first level of the dissection. And in the second three graphs here at the bottom, we have the second dissection between harvest index and total biomass. Harvest index on the x-axis, total biomass on the y-axis. Well, what can we conclude from this? In general, in the population which was grown in the Spanish, in Almeria, Spanish greenhouses, there is a wider distribution of these components, so wider variability, you could say. But for both the Spanish experiment and the Dutch experiment, the yield improvement came more from a higher total fruit dry weight than from the fruit fresh dry weight ratio. And I would say that's good, because if it was from the higher fresh dry weight ratio, that would mean tomatoes which are more diluted with water, and usually that doesn't result in a better taste. And also in both situations we found that the yield improvement is more from a higher total biomass produced than from a higher harvest index. Well, I also explained already we would expect, or we would hope, that we can know better about yield QTLs, where they come from. And I have here one example on chromosome 11. There are two yield QTLs indicated here with 11.1 and 11.2. And then, so the top line is total yield or marketable yield. And then there are the components of the harvest dissection model and the components of the biomass dissection model. And for example, in this one, we can see that this QTL for yield is linked to a higher individual fruit fresh weight and a higher fruit fresh weight per truss. And then very important, how is it with the prediction accuracy? If we predict total yield or marketable yield directly from the QTLs for yield, we have a prediction accuracy of about 0.3, and that is the same accuracy which we can obtain by using one of the three harvest dissection models. Well, the first harvest dissection model is the one I talk mostly about, simply number of fruits multiplied by individual fruit fresh weight, that gives the yield. The other two dissections are also shown here, and in the ellipse like number 2 and 3, but they don't differ in prediction accuracy. Yeah, now you might wonder, what have we gained now? Because the prediction accuracy for directly using QTLs for yield is the same as using the steps in between, with the QTL for predicting the harvest dissection components and then predicting yield. But yeah, we have still the advantages that we know now better where QTLs for yield come from. And perhaps some of these components we can measure in an earlier stage. So it's not necessarily only useful if the prediction accuracy is higher with this approach compared to directly focusing on yield. And we also see that the prediction accuracy for the components, fortunately, that is indeed higher than for yield itself, but you need several components combined to predict yield. So that leads to a lower overall accuracy. Then the conclusions. Well, crop models can bridge the gap between genotype and phenotype. That's no news, I think, by now. Although we started already with that, Fred just remembered me something like 15 years ago. By combining crop models, yield dissection models with QTLs, breeders can design a high-yielding line. I hope I made that also clear, although the time is very limited to make it all clear. And you can focus then on the preferred composition of the component threads, the phenotypic idiotype, and also the assembling positive alleles for component threads, the genotypic idiotype. Well, these component threads lead to more QTLs compared to the yield QTLs directly, and the yield QTLs can be understood. And the last point, these component threads can be determined in an earlier stage, like for example the average fruit size. Perhaps you don't have to grow the tomato crop for 11 months to determine that for a genotype. You can do that earlier. I have two more short slides. If you are interested in this, there's much more to say about this topic. And that's in the thesis, the thick thesis of Yutaka. And it's freely available. If you Google for PhD thesis, Wageningen, Yutaka, probably you will find it. And otherwise, the link is shown here. And that's if you can't wait for the journal papers, because certainly the chapters will be published at a certain moment, but things go a bit slow. Understandable, Yutaka started a new job, so that should get the first priority. And the last slide, you might think, yeah, Eb is all the time talking about crop modeling, but yeah, it's a yield dissection, so that's not a dynamic model. Well, we did this also already for a dynamic model, this approach, so where the climatic data are used over a whole growing season to predict the yield. But there we used simulation data only, so no real measurements from RILs. And to show that that's quite special, Paulo Rodriguez, who did that work in Wageningen, he got a special recognition for that from the journal Crop Science, as you see here. And of course, what has to do with that, the authors, the co-authors are the usual suspects, but there is also Scott. So maybe that makes all the difference. Thank you very much.
Thank you, Eb.